Hello everyone, welcome back. Today I'm going to be going over the epithelial tissue, which is also known as epithelium. So a quick overview. The epithelial tissue or epithelium, they cover body surfaces. You see them forming glands, lining glands. They also line body cavities such as pleural cavities, pericardial cavities, and they form protective barriers. So this is all just general overview, and I discussed in the previous video about their special characteristics in that they are avascular, which means that they do not have any blood vessels. And one of the way they are going to get their nutrients is going to be the tissue surrounding them, the tissue below. So and which also will result in, especially when you have stratification, the top layers are going to be considered dead because they're very far away from blood supply. They're also innervated, they have nerves. We find epithelial tissues on our skin. So for example, if something pokes you or you have a cut, you'll be able to feel it. And they also have a great power, which is they have a regenerative capability. Regeneration is very, very common with epithelial tissue, and I'll talk about them, as it relates to the other types of epithelial tissue. I mentioned also that there are two classes. The way that we categorize epithelial through their shape, and the shapes that I'm going to be talking about today will be squamous, cuboidal, columnar, and transitional. And then we also use layers, such as simple, pseudostratified, and stratified. So I always try to, especially with epithelial tissue, you always have to include the layers and the shape. Just like, for example, everyone has a first and last name, but if you just walk into, let's say, a mall and you say, "Oh, I'm looking for John," well, that's a whole lot of John. Which John are you looking for? You're going to need a last name to basically zero it in. So when you're studying the epithelial tissue, I just don't want you to say, "Oh, this is simple epithelium." Well, is it simple squamous? Is it simple columnar? Is it simple cuboidal? Or I just don't want you to say, "Oh, this is squamous epithelium." Well, is it simple squamous or is it stratified squamous? So you have to be a bit more specific when you're dealing with epithelial tissue. So the first one I'm going to talk about is going to be the simple squamous epithelium. So this has one layer, all right? Single layer. The cells are going to be flat, just like you're going to see here. The cells are going to be flat, and they have a central nuclei. And when you look here, they look more scale-like. So depending on really how you look at it, what we're looking at right now is the simple squamous epithelium in the lungs. And the simple, because it has one layer that we're seeing here, this will allow materials to pass through diffusion and filtration, more diffusion than filtration. So imagine your lungs, you're trying to exchange oxygen for carbon dioxide. You definitely wouldn't want it to go through 20 layers in order for oxygen to get in, or another 20 layers for carbon dioxide to come out. So in this situation, we're going to need just simple squamous. And where do we see simple squamous epithelium? We're going to find them in kidneys. I mentioned filtration. We're going to find them in the lungs. I just gave you an example of that, in the heart, and in blood vessels. Because blood vessels, they're going to have to exchange gases and carry nutrients. So the second one is going to be stratified squamous epithelium. So when you see the word stratified, you're thinking multiple layers. And, when we're talking about stratified, it could be 2 layers. It could be, anything basically more than one. But in this situation, whenever you see stratified squamous, it's definitely going to be more than 2 layers. These are the layers that are stacked. As you can see, they're stacked. They could be from 10 to 20. And anytime you see stratification, protection is key here. You're going to see in areas of wear and tear. You're going to see in areas that are prone to abrasion. So this is going to be the epithelial tissues that's going to be found on your external skin. Our external skin should be able to battle every sort of environmental or, even stuff spilling on you, you should have that layer. And also you'll be able to see that in your tongue, in your mouth area. Imagine you had your hot tea or coffee and, it was too hot and it's going to burn away some of the cells. Thankfully, it's only going to burn off the top layer. It's not going to burn off your the entire layer of your tongue. So you're going to, sometimes we put spicy stuff in our mouth. There's a lot of abrasion going on there. So it needs to have multiple layers. We definitely would not want, just one layer on our tongue, because that could, it, like I mentioned, something got you had a hot tea and then that one layer could be burnt off. And if you remember, the skin, we're constantly shedding our skin cells, so this will let the other layers to basically move up. So we're going to find it in our throat, in our anus, and in the vagina. So the next one is going to be simple cuboidal epithelium. So one of the ways you're going to be able to differentiate this from squamous is that it's going to look cube-like, the word cuboidal, cube-like, and therefore their nuclei, which I'm pointing to right here, their nuclei will be rounder or circular. And another clue to cuboidal is that they're gonna appear in, circular form. They're not gonna be all dispersed like we just saw with the squamous. And here, secretion is going to be more important than absorption. So there are some absorptive capabilities, but secretion is key in cuboidal epithelium. So therefore, we're going to see them in glands, in ducts, in renal tubules. So what we're seeing here is going to be simple cuboidal epithelium in the kidneys. I have here a picture of the thyroid gland. The thyroid gland secretes hormones, so we're going to see simple cuboidal epithelium in the thyroid gland. So the next one is going to be the stratified cuboidal epithelium. The stratified cuboidal, they're not that very common. They're usually 2 layers, unlike squamous. We're not going to have like 20 layers of that. And like I said, again, you're seeing the circular pattern. And we usually find stratified cuboidal epithelium in sweat glands. We find them in mammary ducts, in the breast. So those are the two major places we're going to find stratified cuboidal epithelium. So the other one we're going to talk about is going to be simple columnar epithelium. So if you take a look at this epithelial tissues, you're going to see that the cells appear long, like tall, long columns, and their nuclei is going to be stretched. Unlike the cuboidal, the nuclei is going to be stretched here. The cuboidal is going to be more circular, more round. And another feature or clue that you're looking at a columnar epithelium is going to be microvilli, or is going to be cilia. I'm going to talk about cilia on the next slide. So when you see microvilli, this would let you know that you're looking at columnar. You should not see microvilli or cilia on squamous cell. You're not going to see that on cuboidal. The job of microvilli is to absorb substances. The job of cilia is to move substances. So for example, on your skin, you should not be having that. And another clue is going to be goblet cells. So these white structures that you're looking at are goblet cells that secrete mucus. Again, there is no reason for your skin to be secreting mucus. If it's secreting mucus, that is abnormal. You need to have that checked out, right? So you see goblet cells, then that tells you this definitely cannot be cuboidal. This definitely cannot be squamous. Your sweat glands should be secreting sweat, not mucus. And here we're going to see that absorption is key. Absorption has a bigger role than secretion. So where are we going to find them? We're going to find it in our digestive tract, in our bronchi, in uterine tubes. So the next epithelial tissue I'm going to talk about is the pseudostratified columnar epithelium. So when you're looking at the epithelial tissue here, it looks like columnar because it's obviously not squamous, it's not cuboidal, you don't see that circular pattern, you don't see that scale-like, even if it's stratified. Even if it's stratified, it doesn't look like the stratified squamous. And remember what I said in the previous slide, you see goblet cells, you see cilia, you only find this in columnar epithelium. So when the goblet cell secretes mucus, it's going to be swept by the cilia, which is seen right here. So we usually only see this in the respiratory tract. An example of that is going to be the trachea. So the last epithelial tissue I'm going to discuss is going to be the transitional epithelium. So when you first take a look at it, you're like, okay, it kind of looks like a stratified, but it's really not, because you're looking at the nuclei, right? You're looking at the nuclei, which tells you that it's cuboidal, especially at the tip, at the very top portion, which we call the apical cells. Here we're seeing that the apical cells are relaxed, meaning they appear cube-shaped. So, and then when we look at the bottom, it kind of looks like a little bit elongated. So when we're looking at the apical cells, if they look cube-shaped, which we term relaxed, this actually tells you that this is an empty bladder. However, if the apical cells are flat, then this tells you that it's a full bladder. So basically, the word transitional really means that it changes the shape based on whether you have a full bladder or you have an empty bladder. And the transitional epithelium is also very unique in that you only see it in the urinary system, especially in areas of the bladder, in the organ called the bladder. So that's going to be the end of the epithelial tissue. I hope I was able to, really discuss the epithelial tissue in detail for you. Another thing I wanted to mention is going to be that, I mentioned that you might not be sure when you're looking at transition epithelium. You can also use the process of elimination and say, well, does this have a cilia? Does it have a microvilli? Which it doesn't. Does it have goblet cells? It doesn't. So this is going to be how you're going to eliminate columnar epithelium, whether it is simple columnar epithelium, whether you're looking at pseudostratified, the fact that it doesn't have cilia, it doesn't have goblet cells. This tells you that you're looking at transitional epithelium. Okay, so that's going to be it for today. Thank you. Bye-bye.